is calling us today from Berkeley, California, where he's calling from his house. Before then, a little bit of history of Michael. He was born in Uruguay, but did most of his formal education, certainly in bachelor's and beyond, in Israel. In 2005, he had his bachelor in science in medical sciences from the Hebrew University of Jerusalem. Also received a master's of science a few years after that in medical neurobiology from the same institution. In 2015, received his PhD from under the advisement of Dalit Ben Yosef. The title of his talk was on modeling Fragile X syndrome with human embryonic stem cells. That degree was received in 2015 from Tel Aviv University. He's currently a postdoctoral fellow in Richard Kramer's group, in which he has two primary scientific foci. The first being to study vision restoration using photo switches, novel photo switches implanted into the retina. And the second, which I imagine to comprise the bulk of his talk today, understanding and manipulating strategies to understand retinal degeneration under the effects of retinoic acid and its receptors. He has 12 publications, a number of them in very notable journals such as the Journal of Neuroscience and Neuron, and a patent on his work, also on retinoic acid and its manipulation that was received, I think, just a few years back. Without further ado, I'm looking forward to your talk, Michael, and the floor is yours.
Thank you very much. I'm starting the timer here. Thank you so much for the introduction. You actually are going to save me a lot of time because I was going to tell you a little bit of what I did before I joined the lab of Richard at UC Berkeley. So yes, I was working a lot on human embryonic stem cells and how to differentiate them into neurons. Back in 2009, that was a very new thing. Today is something that everybody can do. But when I finished my PhD, I did a lot of electrophysiology and molecular biology in dissociated neurons, in human neurons from stem cells. And so I wanted to switch gears to working in vivo, to working in mice, to do some more, let's call hardcore neuroscience. And that's how I came to the lab of Richard at UC Berkeley. And that is exactly what I'm going to tell you is my postdoc work and most of what is already published, and a lot of about things that I hope very much to publish in the next year or so. So let's dig in. This is the retina. You guys know it better than me, of course. We're talking about photoreceptor degeneration as it happens in RP, in AMD, in retinal detachment, in photoablation. But obviously, the way we see it in our mouse models, I'm going to talk about the RD1 and the RD10 mouse models mostly. So these mice, they start losing their outer segments of the rods first, then the bodies of the rods are gone and the outer segments of the cones are gone. Finally, you get only some cone somata here and there, and then eventually they will lose all of their photoreceptors. And what is going to happen is that as the outer retina degenerates and these cells are dying, the inner retina is going to undergo a profound transformation that we call overall retinal remodeling, right? And so this might look like a secondary thing, secondary effect downstream to photoreceptor loss, but it's my goal here today to convince you that retinal remodeling is super important. And it's not just another detail of retinal degeneration, but it's a major thing that we really need to focus on, because if we hope to restore vision in some way, and the underlying circuit, the wiring of the inner retina has been corrupted by remodeling, then that restoration of vision is not going to be as successful as we think, right? And so I'm not concerned on saving the photoreceptors. I will not make any attempt of saving the photoreceptors from dying. In fact, I want them to die so I can look at remodeling. And if we look at the mice models, the mouse models that I use, these are the RD1 and the RD10, the fast degenerating, the slow degenerating. As soon as P21 for the RD1 and P45 for the RD10, you have a lot of photoreceptors dead, but many of them are still alive, mostly cones, but even some rods are still alive here and there. And at this early time point in degeneration, you already see the effects of remodeling in the inner retina. You see spontaneous hyperactivity arising in the ganglion cell layer, in the ganglion cells. This is from Stashev, Steven Stashev, 2008. These are MEA recordings, multiple electron array recordings, where you can see that very early in the life of the RD1 mouse, very aggressive mutation, you can see that hyperactivity rising. And another thing that happens concomitant with this at the very early stages of remodeling is that the ganglion cell layers become hyperpermeable. If you dip the retina in a dye that is impermeable to membranes, you see very few cells loading the dye in a wild type retina, but you see 30 to 50% of the cells loading the dye in the RD1 retina, and these are RGCs. You can see their axons here. This is FITC, but it works with many other dyes. If you move forward in time and you let the mouse degenerate for longer periods of time, some other weird things start happening, like there is increased electrical coupling, activation of connexins between amacrine and bipolar, between bipolar and themselves, even between RGCs and themselves. There are synchronous oscillations that happen mostly in off RGCs. These are, the people that work most on these are Peter Detweiler and Thomas Euler, and they have a lot of work on this stuff. They talk about homeostatic plasticity. They talk about presynaptic inputs driving these oscillations. I do not agree, and I will show you some results that show that maybe something else is happening. And then if you wait long enough, really to very late stages of photoreceptor degeneration, you will start seeing that the cells themselves change. The retina itself changes morphologically. There is new dendrites that grow and terminals of bipolar cells that penetrate deeper in the IPL. There is layer disruption. Amacrine cells start invading the ganglion cell layer and so on. The big gurus of morphological remodeling are Brian Jones and Robert Mark from the University of Utah. This is one small example of their work. I want to point out that hyperactivity and hyperpermeability not only happen first, they happen as the photoreceptors are degenerating and they are still there. And they accumulate on time. These other phenomena happen only after we get almost complete loss of photoreceptors, right? And so this is first and persists and accumulates with time. So retinal remodeling, as I said, are a bunch of phenotypes that accumulate with time and start very early in degeneration as soon as the photoreceptors are dying. Remodeling requires death and physical removal of the photoreceptors. So for example, in the triple knockout mouse, where the photoreceptors, the cones and the rods are unable to transduce light, to get stimulated by light, they don't react to light, but they are there. They're alive, and they are not removed from the outer retina. There is no remodeling there, okay? So you need the photoreceptors to physically disappear. And it's universal. We have direct and indirect evidence in mice, rats, rabbits, dogs. William Beltran has evidence in dogs, pigs, and monkeys and humans. So this happens no matter what species you are, and no matter how the photoreceptors die. If they die and they disappear, then the RGCs are going to get remodeled. And I'm focusing on RGCs because the RGCs are the last station before the train leaves, right? I mean, that is where all the information converges on its way to the brain. So if the receptive fields of the RGCs are corrupted, if the wiring of the retina is corrupted, and these guys are getting the wrong signal, then you either catch it there or you don't catch it at all. So that is why my focus is on RGCs. Now, as the photoreceptors are dying, but you still have lingering photoreceptors, mostly cones, that can still react to light, this hyperactivity is going to create a problem because it's basically noise that is going to cause the signals to be swallowed in the noise. It's a signal to noise ratio problem, a typical signal to noise ratio problem. And after all of the photoreceptors are dead, you still have the problem, right? If you want to now restore vision with prosthetics, with optogenetics, with photopharmacology, the famous photo switches that Richard is famous for, you still have this noise. So we need to address the problem if we hope to improve vision of people that have either early degeneration or partial degeneration. And if we want to restore vision in those that have complete degeneration like retinitis pigmentosa, we also need to address this problem. So I hope that I convince you that this is very important. And so I'm going to focus on the hyperactivity and the hyperpermeability. And I'm going to ask what drives these two phenomena here that are first, that are early, and then persist for the longest time. Well, if you look at the literature, you will see that Brian Jones and Robert Mark talked about the involvement of retinoids in the morphological changes that you see very, very late. And if you look at these phenomena, these oscillations and this increased electrical coupling, then the explanation here that is proposed is usually maladaptive plasticity, some type of aberrant homeostatic plasticity involving glutamate and GABA and so on. And also another explanation is the upregulation of connexins that create this network of electrical activation throughout the inner retina that is going to distort the receptive fields of the RGCs. We wanted to focus on this, and we wanted to ask what is driving this. So let's look at these two phenomena and how we measure them, okay? So let's begin with hyperactivity. This is how we look at hyperactivity. MEA, multiple electrode arrays, lights on, lights off, lights on, lights off, RD10 mouse, P14, very young, still intact retina, everything is fine. You see on responses, off responses, and everything looks quite nice. As the mouse ages, these responses start dwindling, and you can see the baseline activity going up. And when the responses are almost completely gone, there might be some cones here and there, but it's very blind. Then you see this huge increase in the background noise. And the two phenomena follow an opposite pathway, right? As the responses dwindle, the background activity increases. And of course, in the RD1 mouse, the increase is even faster because the degeneration is faster. But here is a crucial point about hyperactivity that I want you to remember. And is that as opposed to what other people say, which is that everything is driven by presynaptic inputs from bipolar and amacrine cells, hyperactivity, early and persistent hyperactivity of the degenerated retina is completely intrinsic to the RGCs. Here we block all of the inputs, metabotropic glutamate receptors, ionotropic glutamate receptors, GABA, glycine, acetylcholine, dopamine. We block all of them and the hyperactivity persists. We are, of course, recording from the RGCs, right? The retina is facing down on top of the electrodes. And so this happens in the RGCs inside of them. This is not mediated by any neurotransmitter that we know of. That gives us a clue that it might be some other type of signal that we need to get. And when we look at permeability, the permeability is not functionally something that is going to cause any loss of vision, as far as we know. But the issue is that it's really easy to model, it's really easy to measure. You just dip the retina in the dye and then look at the cells and count the cells. And you can see the difference between the wild type and the RD1 retina. And previously, this is a paper that I wasn't the lead author, but I was a part of it, where we showed that this hyperpermeability is mediated by the activation of a group of purinergic receptors called P2X, and specifically the P2X7 isoform, which is the most upregulated, transcriptionally speaking, when you compare RD1 to wild-type retina. And this is happening specifically in off RGCs. The cells that are peaking the dye are mostly off RGCs, okay? So let's keep those two things in mind. Hyperactivity, which is intrinsic to RGCs, and hyperpermeability, which is mediated by P2X7 and happens mostly in off RGCs. And so there, as I said before, there must be a signal that it tells the RGCs, hey, the guys upstairs, the photoreceptors are dead. Now please go crazy. And we took our cues from the work of Brian Jones and Robert Mark and from other studies. And we also looked at the possibility that retinoids might be involved in this phenomena. Retinoids in the retina follow a very known metabolic pathway. Retinol becomes retinaldehyde, which becomes retinoic acid, which activates its receptor, which is a nuclear receptor that is in charge of transcribing genes that are downstream to it. And the way that RAR does what it does is because it binds to specific sequences in the genome known as rare retinoic acid response elements, which are different sequences that you can use as rares. And if you take those sequences, I took that and I cloned it in a construct that has GFP and downstream to those rares with a weak promoter in between them and another fluorescent protein with a constitutive expression just to see where the construct goes. And you see here in hex cells how when you don't have retinoic acid in the system, you only have RFP. But if you add all trans retinoic acid, you see the GFP emerging. And so you pack this into a virus, infect the retinas of mice, and you can see, you infect the retinas, you take them out ex vivo, you look at the RGCs, and you see a lot more green cells that are also greener in that mouse. This is a mouse that has a mutation in PD6 beta. Here is a rat, so a different species, with a mutation in rhodopsin, a different mutation, a different gene, and still the phenomenon is the same, right? You get death of the photoreceptors, you get activation of RAR in the retina, in the ganglion cell layer specifically. And also we proved, I'm not gonna show it here, but we proved that these are not astrocytes by doing some immunohistochemistry work. And now the question is, okay, so there is more RAR activation in the ganglion cell layer when the mouse goes blind, when the photoreceptors die in the outer retina. Is that necessary and sufficient to induce remodeling? So as I told you before, hyperpermeability is easy to model. So if you inject a wild type mouse with ATRA, you will see how the cells become hyperpermeable, resembling the RD1. If you take the RD1 and you inject the mouse with BMS 493, an antagonist of RAR, you will see how they become non-permeable, like the wild type control. This works with drugs, but also works with genes. This is a dominant positive construct of the retinoic acid receptor that I packed into a virus and introduced into wild type mice. You see every single cell loading up the dye. And if you take a dominant negative retinoic acid receptor that is truncated and cannot be de-repressed, and you infect RD1 retinas with that, you see the permeability going away completely. These cells that are still picking up the dye, by the way, they are pericytes of the vasculature. We don't know why they pick the dye, but they do. Most importantly, from a functional point of view, is that whether you block with the drug or with the gene, you get downregulation, significant 50% reduction in the hyperactivity. And this is crucial from a functional point of view, because if you take now a mouse that is only half blind, like the RD10 mouse at mid-degeneration point, like P50, let's say, something like that, now you inject one eye with vehicle, one eye with the BMS, the antagonist for retinoic acid receptor, and you shine the same flashlight at the same intensity, the same duration. You can see how in the drug-injected retina, a signal, a response will emerge from within the noise because we have reduced the noise, while in the vehicle control, it doesn't happen. These experiments were done in a double-blind setting, and in 5 out of 5 mice, every eye that was injected with BMS responded better than every eye that was injected with PBS, the vehicle. And this is not done on one single intensity of light. We do it with many different intensities of light. And of course, when the light is too strong, the response can overcome the noise by itself. It's actually with the dim light or the light that is less intense that you need help to overcome the noise. And that's exactly what we are doing here. So this actually, again, this proves that the problem here is a signal to noise ratio issue that we are changing by suppressing RAR activity. And this is all ex vivo and is great, but let's see what happens in vivo. Sorry, that was ex vivo, right? MEA recordings. In vivo, what we did in that paper was 2 different behavioral paradigms, very simple. The first one is this double chamber box, which, one chamber is dark and the other one is either dark or with light. You turn on the lights you turn you turn off the lights, the mice will spend 50% of the time in either chamber. An infrared sensor will measure how much time they spend on either side. If you turn the light very, very strongly, they will move into the dark chamber. But if you turn on the light at an intensity that is 10 times lower, only the mice that got the dominant negative gene, the dominant negative version of RAR, will move into the dark chamber, and the ones that were untreated just don't see the difference between darkness and this dim light. A second paradigm that we use is fear conditioning. So you just give the mice a shock while pairing it with a very bright light. And then you do that 3 times, then you come back the next day and you just shine the light without delivering the shock, and a camera will track the movement of the mouse, and the mice will just freeze because they anticipate the shock. And then you measure the percent time freezing, and you can see here very simple that the behavior of the mice that were treated resembles a lot more those that of the wild type with good vision than the RD10 that weren't treated with bad vision. But light perception is not vision, okay? So I'm going to talk about vision in a moment. Just keep that in mind. Light perception is not vision. So this is the summary here, interim summary. As you go blind, as your photoreceptors are dying, and obviously after they're completely dead, you have activation of RAR in your RGCs, maybe also in bipolar cells, maybe in amacrine cells, I don't know, but I'm looking at RGCs, and that creates intrinsic hyperactivity in the RGCs, which will interfere with whatever signal you want to pass, whether it's a signal coming from the lingering photoreceptors or from some restoration of vision by some technology. But if we block RAR, we reduce the noise, we allow the signals to go through. And so this is what we think is happening, right? Think about this. In the mouse, 70% of the retina is photoreceptors. All of the inner retina, the bipolar, the amacrine, the RGCs, everything else is just 30%. And the RGCs, in fact, are the minority of the minority. So you can think about the retina of the mouse, at least, and probably that's true for humans, as a big sack full of retinaldehyde, because every single photoreceptor is packed with opsins that need retinaldehyde. Now, these cells are dying, and they don't die by apoptosis, they die by necrosis. Their contents are physically spilled out. So there is like popping balloons, right? The retinaldehyde is just popping out and coming out of the cells as they are dying, and it has to go somewhere. And probably the RPE is still providing vitamin A and retinaldehyde, recycling it, because the RPE doesn't know that the photoreceptors are dead. So all of that huge amount of retinaldehyde has to go somewhere, and it eventually arrives somehow into the inner retina, where through Muir glial cells or other retinal neurons is converted at the end of the day into retinoic acid, because RALDH, the enzyme that converts retinaldehyde to retinoic acid, is everywhere. And then RAR will transcribe genes. We know that for heightened memory permeability, it happens through P2X receptors. About the heightened spontaneous firing, we still don't know. Maybe some voltage-gated channels, maybe some ionotropic receptors that get activated somehow. That is still speculation. Okay, but this is the working model right now. Okay, just to be clear, because I know that these are very aggressive mutations in mice that resemble RP, which is a rare disease, we ask what happens when there is no mutation and when the death of the photoreceptors is localized. And so we partner with Daniel Palanker from Stanford, and we introduce a foreign object subretinally, a disc of an aluminum disc, into the outer retina, which kills only the photoreceptors that it comes in contact with, and the others remain alive, right? And then you look at the RGCs downstream or underneath the area of the lesion versus the RGCs far away. And using the reporter that I invented, I did the same thing, the same strategy. And again, only in the area that is degenerated, you see the GFP coming up. And in the control areas, you see very little GFP. And that happened in every single rat that we looked at. Using the permeability assay, this loading dye, you can even look at the dispersal of the retinoic acid signal from the epicenter of the lesion outwards, and we measure it to be about 250 to 300 microns away, you stop seeing this hyperpermeability that we know is caused by retinoic acid. And just for good measures, also the hyperactivity is local. The hyperactivity, which is still intrinsic to the RGCs, will be seen only in the degenerated area and its vicinity and not farther away. So everything works the same. As I said many times, it doesn't matter how the photoreceptors get killed and how much they get killed. The moment you have degeneration in the outer retina, you have remodeling in the inner retina in the RGCs. So I hope I convinced you that this is an important phenomenon that is worth our research. So now let's move to some new questions and some recent ongoing research that I hope to publish soon. Let's start with the most recent one, which I only have one figure to show you, and is about whether RAR is the trigger also behind other aspects of remodeling, right? Remember, okay, I show you this is caused by RAR, right? We are pretty sure that works. Brian Jones and Robert Mark tell us that retinoids are involved in this stage. What about right in the middle? What about these weird oscillations? So I am an electrophysiologist. I do patch clamp. That's most of what I do, so we can patch onto alpha cells because we can recognize them anatomically speaking. Then we have an intracellular dye in the patch pipette, so we can measure the stratification of the dendrites and say this cell is on, this cell is off. And this I've been doing this with Scott Nowie, which is who is a visiting professor at our lab, and I'm sure some of you know him. And so when we patch onto off alpha RGCs, right, and we hold them at 0 millivolt, we see the same exact oscillations that Margolis et al. 2008 saw. The same thing, right? However, if you now go back to these cells in a mouse that has been injected with BMS-493, this RAR antagonist, the oscillations are gone. So this is a very new stuff that we just started a few months ago, and we are accumulating more and more data. And I hope it will go into the direction that I think it's going, which is explaining that even if there is a maladaptive plasticity, it might be, again, because of RAR, right? I mean, our hypothesis doesn't necessarily deny their hypothesis. The two things might be working in synergy. Now let's move to the more clinically oriented stuff. This is something that I really like to do, and is, trying to translate this into something that we can use for people. And the question here is, can RAR inhibition improve visual acuity? Because I showed you, remember, I showed you those behaviors. That was light perception. Light perception is not vision. And so to measure vision, we designed a very new and I think a really great tool. This is an operant conditioning cage that I bought from Lafayette, and I started playing with it. And what we did is we created a situation in which the mouse can run only in one direction, which is focusing the attention of the mouse on a screen that is on the back of the cage and is controlled by a mini computer, a Raspberry Pi. And then the mouse is trying to recognize the transition from a blank image to vertical stripes image. And then that is the cue for the mouse to leave the wheel, poke through the reservoir area where there are infrared beams, and then get the reward, which is 10% sucrose, right? And we measure the latency, we measure the success rate, how many times the mouse did it right, but we also measure mostly the latency, which is how long it takes from the moment the image appears to the moment the mouse pokes through the infrared sensors in the reward area. And so a young RD10 that has most of its vision pretty intact, P35, P40, it's seeing, okay, sees the images, boom, leaves the wheel and gets the reward, right? The same mouse gets older, like we all do, and it gets pretty blind. And he runs because they remember the task. I mean, we did that in wild type. They remember the task. But this mouse is just too blind. The image doesn't tell him anything. He keeps running and he misses his opportunity to get the reward. So the latency will be timeout after 10 seconds, right? And so we can train them within 3 to 4 days. This is a training session in a young RD10 mouse. You can see at the beginning of the night, the mouse is still trying to guess, but by the end of that night, it's 12 hours from 6 p.m. to 6 a.m. By the end of that night, the mouse gets it. And they keep going. Even if you make the test more stringent, you reduce the timeout to 10 seconds only, they just go for it. But as I showed you before, if you wait long enough, they become very, very blind. They are missing a lot of pokes. And those pokes that are successful are just by chance, because you can see the huge standard deviation here as compared to the almost non-existent standard deviation here, right? And then we can now take the same test, but instead of showing either blank or vertical stripes at 100% contrast, we can randomize every trial to show a different image with a different contrast. And in that way, we are going to measure the latency for every single contrast, again, in a randomized way. And we are able to create a contrast sensitivity curve that in wild-type animals looks very nice. Already at 8% contrast, the mice are responding, and they basically plateau after 16 or 32%, okay? And these are wild-type animals with healthy retinas. Of course, RD10 mice, even if you catch them at P40 and they have pretty good vision, it's still not the same as a wild type. They still have some degeneration going on. So their baseline activity doesn't look as nice as the wild type, but they are seeing, they're seeing something. Now we wait 30 more days and we can inject the mice with vehicle control and see that they are basically completely blind and not responding to anything. But if you now inject another group, a counterpart group with BMS intravitreally a few days before the test, you see that even though their vision has is very degraded, but it's still much better than the controls, right? So there is some preservation of vision that is going on here. Now, BMS needs to be injected in the retina. It's messy, there is backflow. You never know how much drug is there. It's a very toxic drug. We don't know its pharmacodynamics, its pharmacokinetics. So we looked for something else, something that is more clinically relevant, because as I told you before, this wants to be a preclinical work, basically. So we found disulfiram. This is an ALDH blocker that is given to alcoholics because it blocks ALDH, which catabolizes the conversion of acetaldehyde into acetic acid. So if you're drinking alcohol and having disulfiram in your system, you're going to puke and you're going to feel horrible, and that discourages people from drinking. It has many disadvantages, of course. This is a drug that you cannot take for long periods of time because it's toxic to your liver, but it crosses the blood-brain barrier. It is FDA approved. It's given per os. And if you don't give the mice alcohol, I hope they're not, getting some whiskey somewhere, then they're fine. They should be fine. And so we combine the food with we combine the drug with food, and we just feed the mice constantly. You can see here that the drug here, this drug works the same way that I showed you with the RDN and with BMS. It reduces retinoic acid in the retina, even though it is given through per os, right? This is the reporter in the RGCs of mice with control or disulfiram diet, and you see the reduction. And it also reduces hyperactivity, similar to what we saw in RD1 mice with BMS 493. Here are RD10 mice, almost completely degenerated with like 20 or 30 days of disulfiram treatment. They also get their hyperactivity in the RGCs reduced. And in the behavioral test, which is what we really are after, we can see again this baseline that is not beautiful, but it is what it is. And then 20 days later, we do the baseline, then we separate the mice into groups, of course. These get disulfiram in the food, and the others get just normal food, regular food. And you see that 20 days later, the guys that are eating disulfiram look better than those that are not eating disulfiram. And 40 days later, the difference is even more emphasized, right? These guys here in the black squares are pretty blind, and the red squares, the disulfiram treatment, are in a better situation. Again, you are not saving the photoreceptors, right? So the mice are going blind. We are helping them see better for whatever time they still have until all of the photoreceptors are completely dead. So this will be an improvement of vision for people that have partial degeneration, or conversely, early stages RP, right? And so based on these results, I hope very much that by early next year, maybe a little bit later, we're going to start a clinical trial with our collaborators in Israel at the Shiva Medical Center, Dr. Igal Rothenstach. He will give disulfiram to RP patients in early and late stages that will be randomized, and will be crossover a clinical trial. Every patient will get the drug and the placebo with a washout period in between them, and they will be randomized. We will look at, at vision, whether they actually see better or not. We will look at microperimetry to see if the scotoma has changed or not. ERG, there's something now called the C-wave, where you actually maybe can track the activity of the RGCs themselves. But another thing that is very interesting is this hyperactivity in the retina the source of the photopsias and the phosphines and these flashlights that people that are going blind see? Right? I mean, it follows that one must be connected to the other, but we need evidence that it is the case. If you read Stephen Stachev's paper from 2018, he is pretty convinced that that is the case, but still we need the evidence. So this clinical trial will, on one hand, ask whether disulfiram is helpful for these people, but we can also ask some basic science questions that will be nice to get the answers to. Okay, so I'm moving forward. And now in this case, moving forward means moving downwards. I'm going to leave the in vivo stuff and the behavior and the preclinical studies and go back to the science, to the fundamentals of what is going on in these retinas, and specifically in the RGCs, when RAR becomes hyperactivated and causes the spontaneous firing, the hyperpermeability, and so on. Now, I told you at the beginning of the talk that when it comes to hyperpermeability, the culprit here is P2X7, and we established that very, very nicely doing a lot of pharmacology and some genetic engineering. And so I knew that the immediate effector of RAR for hyperpermeability is P2X7. I asked, what else is P2X7 doing? And to do that, what I did is I just created a new double transgenic mouse, a double mutant mouse, which is RD1, so it's going to lose all of the photoreceptors pretty soon, but it's also P2X7 knockout. And you can see here, this is qPCR, this is, sorry, not qPCR, this is RT-PCR, reverse transcriptase PCR from RNA. You see there is no expression of P2X7. And when you load the dyes that are impermeable to retinas, you see in the ganglion cell layer and in the inner nuclear layer what you expect to see, right? No loading besides the pericytes in the wild type, a lot of loading in the RD1, and nothing in the P2X7 knockout, right? Again, these are the pericytes, so those are not the cells that we are interested in. That is just something that we expect, and that is to confirm that it really works. What we weren't expecting is that P2X7 is also necessary and sufficient to induce the intrinsic RGC hyperactivity. If you put these retinas on the MEA and compare them to the RD1 with P2X7, you see that the hyperactivity has been cut by half or even more. What is interesting about it is that if you block P2X receptors acutely with a drug, you perfuse the retina for like 15, 20 minutes, we block the permeability, of course, but we don't block the hyperactivity. It's only when you remove P2X7 from the genome, when you knock it out, that you get this reduction. And again, it's intrinsic to the RGCs because it's the same whether it's in selling or in selling plus synaptic blockers. Furthermore, if I now take a construct delivering P2X7 under the expression of the chicken beta actin promoter, which is a very aggressive promoter that causes overexpression of P2X7 in this case, we deliver that into wild-type mice. Now you see hyperactivity rising in the wild-type mice. That is very, very quiet usually, right? It's always around 0.5 to 1 hertz. You get 2, 3, 4 hertz of activity, and there is some maintenance of this activity even when you block all of the synaptic inputs. So this shows that P2X7 is doing something else, not just opening the pore so these molecules can go in, of course. there is influx. We think there is influx of calcium. That calcium influx is tonic. Basically, it's chronic to the cells, and the cells will start reacting to it in a kind of snowball that will eventually lead to the hyperactivity. Now, I always go back to the patch clamp because it's a pain in the it's a pain to do, but there you can get so much information from doing patch clamp. If you have the patience to do it and to patch every single cell and you accumulate data, you will see a whole picture eventually that is really, there is no technique like it, that nothing can replace it, I think. And so, as I told you before, we patch on cells without knowing if they're on or off, but we can know they're on or off after the recording because we can see this intracellular dye going into the dendrites. And just by doing a simple IV curve, you can see that in honor GCs, nothing basically changes from wild type to RD1 to RD1 P2X7 knockout. They look the same. But if you look at the offer GCs, and remember, these are the cells that are also hyperpermeable, and so we think that the P2X7 expression is mostly there. Here, what you can see is that in the hyperpolarizatory side of the curve, you have much larger currents in the RD1 that are completely knocked out by knocking out P2X7, pardon the pun. So there is an effect, there is a physiological effect of P2X7 that involves some hyperpolarization activated signal. And who is that? Well, that is most probably HCN. HCN channels are hyperpolarization activated, cyclic nucleotide gated channels. They are also called the funny current because they are activated by hyperpolarization, but then they cause depolarization, right? They're very important in the heart, for example, in the beating of the heart, in any rhythmic activity, they are very important, but they're also expressed in the brain and they are also expressed in the retina. When you measure the density of the IH current, of the HCN current, as a function of how deep the dendrites of the RGCs go into the INL, into the IPL, meaning on cells versus off cells, or putative off and putative on, if you will, you see there is a tendency for higher IH in the off RGCs, but that becomes really increased when you have degeneration, right? The RD1, almost all of the off RGCs have a lot more IH. And that is completely demised when you knock out P2X7. So RAR is upstream to P2X7, and P2X7 must be upstream to HCN. When I run some immunohistochemistry experiments on this, I look at HCN4 and I didn't see any change. But then when I looked at HCN1, I saw a big change. You see how HCN1 is found mostly around the somata of the bipolar cells. And then this huge, really bright band in the center of the IPL, right in the center. And when you knock out P2X7 and you look at HCN1, you see that it remains in the bipolar cells, but actually that specific central band in the IPL is gone. And we know that this is transcriptionally mediated because if you, this is qPCR, this is real-time qPCR, you extract RNA from retinas. I always compare RD1 to RD1. I never compare RD1 to wild type because the cellular content is very different, right? The wild type is mostly photoreceptors. So you're not looking at the RNA of the same type of cell. So that is not kosher. You want to look at RD1 versus RD1. So here, RD1 versus RD1 P2X7 knockout, you look at all four isoforms, and you can see that HCN1 and a little bit also HCN2 are the most one that get downregulated when you knock out P2X7, and not so much HCN3 and HCN4. So this is not a protein-protein interaction. I mean, even though P2X7 is in the membrane and HCN is in the membrane too, they are not interacting directly with each other as far as we know. This is happening through some mechanism that involves some signal that gets HCN upregulated at the transcription level. So HCN is downstream to P2X7, which is downstream to RAR. So we need to reform our model. Our model is not correct, right? I showed you this before, but this is not right. Now we know that this is not working. What really happens is this, right? So from RAR, we go to P2X7, which through some signal is upregulating HCN, which probably has something to do with the hyperactivity. That is the part of the work that we are trying to determine. Now we're trying to determine two things: whether this is really mediated by the influx of calcium, and whether this is really correlated to the heightened spontaneous firing that you see at the MEA level. There is a problem with that, which has to do with the drugs that block HCN. They're very messy and they block other things. They're not very specific for HCN. So that is part of the technical issues that we need to think about. But I'm very hopeful that this will come to a more clear picture. I don't think that we are going to get every single detail, but we will have some more mechanistic insight in the near future. And that's it. Thank you very much, everybody. Thank you for listening. This is Richard, my supervisor and mentor, that I love to fight with him, and he loves to fight with me, I hope. And Bristol and Zach were essential for this work. Daniel is an amazing technician that is helping with the behavior, because waking up every morning at 5 a.m. to be in the lab at 6 a.m. and doing that every day is a little bad for your health. So he's taking some of that work upon himself. And these are the papers. I will be obviously glad to share them with you. And that's it. Thank you very much.
So I'm happy to moderate the chat if anyone has any questions, but I think we have a tight enough group here that if you want to unmute yourself, please ask questions directly to Michael.
Hi, Michael. It's Dave DiLoretto here. What do you think the role of the glial cells are in this process? Is the retinoic acid completely from breakdown of the photoreceptors, or is gliosis and upregulation of the production of this part of the process?
Yeah, that's a great question. I really don't know one way or another. There are papers saying that vitamin A can come into the inner retina through the vasculature, actually. So maybe, as we suspect, anatomically speaking, the Müller glial cells span the whole retina. So they are the #1 candidate to be those that pick up either retinol or retinaldehyde and then bring it down to the inner retina. But we don't really know that. And we want to, I was looking for a long time for a way of killing the Müller glial cells to see that the hyperactivity goes away. And there is no really good way of doing that. They seal very fast. I thought about killing them with a laser, but it doesn't work. The other option was to make a very complicated genetic engineering process that we couldn't do. So yeah, it's a very interesting thing. But I will tell you one more thing that is interesting. The glia really encapsulate the whole inner retina. When you patch onto the RGCs, there's actually a membrane that you need to break. Mm-hmm. And that is the end fit of the Müller glial cells encapsulating the inner retina from the anterior side. In the RD1P2X7 knockout, that membrane you can break it super easily. It's like, it's not even there. So I don't know. Remember, the P2X7 knockout is global knockout. It's not just in the RGCs. I'm looking at the RGCs, but it's global. So yeah, there must be some involvement of the Müller glial cells, but I really don't know.
Did you look at the histology, the difference by blocking with the disulfiram, if there was a histological decrease in gliosis versus the retinas that were not blocked?
So in our histological assays, we just measure apoptosis because we want to know if the drug is toxic to the cells. So we cannot see actually gliosis, but that will be a good idea.
Thank you.
Thank you.
Hi, Michael. I enjoyed seeing your work and the updates that you've added to it. Question, and I may not be able to phrase this quite right, but so a lot of the work's done in rodents, but when we move to human and primate, there's the offset between the photoreceptors and the ganglion cell layer. And so do you, or does anyone have evidence, is the upregulation of retinoic acid, is that going to be if the photoreceptors here are lost, so, I'm thinking regional losses or localized or photoreceptor loss. If it's lost here, is that going to then impact the ganglion cells directly above? Are those the ones that are remodeling, or is it the ones that these photoreceptors would have been inputting to that are displaced, the ones that are remodeling? And where does retinoic acid play in that?
Right. So if it worked, if it worked by synaptic inputs, then the second option would be what I would bet on. But actually, what we show, and I think this is, these results were very, very clear here. We saw this in every single animal that we treated, is that there is some dispersal of the signal from the degenerated area outwards, but it's mostly in the degenerated area, right? So it really decays with time. So I think that if I could test this in humans, I would say that it will, as far as I know, it mimics what we are seeing here, because this is the evidence that we have. I think that the retinoic acid is just moving freely. It's a very lipophilic molecule, and is either being carried or is moving freely and is penetrating wherever there is an opening for it to pour in. So that is what I would put my money on. But yes, of course, that is something that we would like to test, right?
Right. I mean, that could be potentially advantageous, taking advantage of that offset in a way similar to what Bill's been doing with Dave with the sort of the GCAMP, the foveal ring, but potentially taking advantage of that offset for tracking directly above photoreceptors or control locations without photoreceptor loss.
Right. Yeah, absolutely. And tomorrow in the Chalk Talk, I'm gonna talk about that exactly, and some things that we've been talking about on how to make lesions and try to find retinoic acid in correlation with those lesions. Yeah, absolutely. No more questions?
So Michael, sorry to be butting in here. I could just send you an email about this, but here's one of the things that I'm wondering about. You showed that disulfiram and the BME are pretty effective at decreasing interretinal noise. And so what I'm wondering about is that you're at sort of a transitional period where you're losing a lot of photoreceptors. And so part of what's going on is the retinoic acid in the inner retina caused by this loss, and part of what's going on is the remodeling. And it's tricky to tease those apart. And you showed pretty clearly that at a mid stage and even at a pretty late stage in degeneration, you can improve things. So how do you deal with that?
So yeah, so that is, yeah, I think that I know what you're meaning here. So what happens after all of the photoreceptors die? Then the retinoic acid should disappear.
Yep.
Right?
Yep.
So that is exactly what we're doing here. We're wrapping up a paper with Bristol that has been, a partner in crime, and we took RD10 and RD1 mice. This is something that I didn't show you because I didn't have time. And we inject them with the virus. So the virus, the same reporter that you can put in the retina, you can inject in neonates in the tail vein using a different serotype called AAV9, developed by David Schaffer and John Flannery. And then you can track a group of mice as they get old. Mm-hmm. And you can see here, for example, at 4 months old, way after all of the photoreceptors are completely gone in both mice, especially the RD1, you still see hyperactivation of RAR. And my conclusion from that is that the RPE, as I said during the talk at some point, the RPE doesn't know the photoreceptors are dead. So it keeps providing either retinol, vitamin A, or recycled retinol from retinolide or whatever, but it keeps putting out retinoids for the photoreceptors that it thinks are still there, but then those retinoids have nowhere to go.
Right.
But one of the challenges—
6 months, this is really decreased, indicating that the few photoreceptors that are left are still important to what's going on.
Yeah, absolutely. No, yeah, I agree with that. Yes, totally. Yeah. Now, why does it decrease so strongly at 6 months of age? Well, maybe we think that, and this is something, again, something that I didn't have time to talk about. Eventually, if you wait long enough with remodeling, the ganglion cells themselves will start dying.
Right.
So that might be part of this. It might be part that the ganglion cells somehow adapt to it and react. I don't know. Yeah.
Michael, I sort of alluded from your talk that most of these effects are happening through, RD1 and RD10 mice are, just this massive atrophy of outer retina, and that's presumed to be, I guess, scotopic function, mostly rod function. I'm wondering, are your models interested in the future looking at specifically cone degeneration? And do you think that this is going to be equally applicable to sort of the photopic regime, or is this just a scotopic phenomenon?
Right. So first of all, let me explain something here. The mutation is in the rods, and they die first. And all of the vision that we get through all of these behaviors that we run is all photopic. It's all cone mediated, because those are the cells that you still have. So let's make that clear. Now, the cones die because the rods secrete as a survival factor that keeps the cones alive. And because they are dying, then the cones don't get that survival factor, and they eventually die too. Those are the mutations that we have. I was thinking about eventually using albino mice, in which you get the cells, you get a perfect healthy retina if you keep the mice in a room that is not very bright. But then when you expose them to bright light, they start undergoing degeneration, and there the cones die together with the rods. So it's a different type of degeneration. I don't know if that answers your question.
Yeah, so albino mice are one, but other specific knockouts that are looking at, cone specific knockouts, do you think the same mechanism is driving degeneration in maybe a cone dominant form of degeneration?
I would say yes, but I need to test. I need to try it. Yes, absolutely. Yeah. I think, again, there is evidence from dogs, there is evidence from rabbits. In every single of these these animal models, you get a different type of degeneration, but you still see remodeling in the inner retina. So I would guess that yes.
Great. Other questions? If not, we're just about at time. Thanks again, Michael, for a wonderful talk. Enjoy the rest of the one-on-one interviews today, and look forward to your Chalk Talk. A quick reminder to everyone, especially the faculty members, Michael's Chalk Talk will be tomorrow at 12 o'clock. You'll see the link in a separate email. Thank you so much. Enjoy your stay, Michael.
Thank you. Thank you very much.
Thanks, Michael. Great talk.
For coming. Thank you.
Yeah.